So welcome to this NAA webinar, Getting the Timing Right, Plant Phrenology and Mismatch in Our Changing Forests. So my name is Maura McGinty-Kloss. I am the Director of Programs and Communications, and I am delighted that you are able to join us today. The Natural Areas Association, if you haven't heard about us, also called NAA, is a national nonprofit organization that serves practitioners who manage ecologically significant landscapes to protect biodiversity. NAA provides applied science, practical solutions, and I've just lost my cursor. Here we go. Practical solutions and knowledge transfer related to the management of natural areas. And I'd like to add, did you know that NAA has enhanced our YouTube channel? We've had YouTube for a while, but a whole lot of great on-demand quality conservation science programs like this webinar today have been added. So please go to our website if you want to use this QR code and check it out. Or if you're already subscribed to YouTube, just search Natural Areas Association. And I think you'll find some great on-demand programming. Mason Heberling is the Associate Curator of Botany at Carnegie Museum of Natural History. He's a botany curator and plant ecologist studying the functional ecology of understory plant species in temperate forests, especially in the context of climate change and introduced species invasions. As a museum curator, he seeks to facilitate and broaden the use of natural history collections by students, researchers, and the public. As a museum-based researcher, he uses the herbarium specimens, field experiments, and observational data to understand basic plant function and complex ecological interactions. His research program explores a diverse range of topics under the umbrella of global change, biology, and museum collections. So Mason, I'm going to let you take it over from here.
Yes, hello. Thanks so much for coming here today. Despite you might be have a lot of field work or other things going on, so I really appreciate you being here, and I'm excited to give the talk. I was eagerly watching to see some familiar names and some not familiar names in the participant list, so that's cool. So yeah, let me just jump right in. So yeah, I'm Mason Heberling. I'm the Associate Curator of Botany at Carnegie Museum of Natural History here in Pittsburgh, Pennsylvania. And I just wanted to kind of give a little pitch of kind of who we are. I love my job. It's very unique in many ways. And I get the opportunity to work with students kind of very much like a professor does, but then also like hands-on and a lot of outreach and museum outreach, working with exhibitions, et cetera. And then also have the privilege of kind of stewarding the Herbarium collection here, which is quite a privilege and honor and an inspiring place to be. Really at the core of all that we do is the Herbarium. And I wanted to give a quick pitch to my fellow botanist colleague, collection manager here at the museum, Bonnie Isaac, who's inspired a lot of the stuff, especially a lot of the Herbarium work that I do. And then I also wanted to give a quick shout out, so I don't forget, to my collaborator who has worked very closely on all the phenology things I'm going to talk about here today, and that's Sarah Kibbing, who was at University of Pittsburgh just across the street and recently moved a little bit farther to Yale University, and together we have a National Science Foundation grant, and a lot of the science that I'll be talking about today is a result of that project. So I'd like to kick off with this kind of beautiful picture of red trillium here in my backyard. I've always been fascinated kind of by the spring, and I'm guessing a lot of people enjoy spring, especially in the temperate Eastern North America in particular. And it's such a short time now, and especially as we're all so busy doing different things, or it's easy to miss, especially if you're teaching classes or just otherwise occupied. It's really easy to miss. And next thing you know, summer's here and you missed a lot of fun plants. And so a lot of my research is thinking about this early springtime and a lot of kind of unexpected implications and importance of spring. I'm guessing a lot of people kind of tuning in also have recognized a lot about spring. And really, when you pay close attention, there's a lot going on in the understory. And oftentimes it's very ephemeral, very quick. If you're off for a week, you might miss a lot going on. And it's really this critical window of opportunity. And that's a lot of what I'm going to be talking about today. And not only the understory, but also the overstory. I can't help myself, but as I was putting this together, I was kind of taking slides from an older talk, kind of in the early days of giving online talks on this topic, kind of in the days of the early pandemic during the shutdown. And a little bit of Pennsylvania humor I can't help is there was a governor order that basically the beaches of Pennsylvania, any of the beaches were closed. However, American Beach didn't follow that in 2020, and in fact opened its leaves in May 15th. And this is a webinar, but I'm assuming people are really rolling on the floor laughing at this. So yeah. So thinking about, we know our forests are changing in many different ways, and kind of this kind of fundamental question behind a lot of my current research in the phenology realm is, are phenological strategies also disrupted? What are the impacts? What are the consequences of differing phenologies and differing responses to phenology? And this really it really wasn't a part of my research actually until maybe 5 years ago or so. And it kind of actually was inspired in part by some of the work in an exhibit here at the museum and working with the collections, thinking about climate change affecting phenology, insights from museum specimens. Here's spice bush, common shrub in Pennsylvania and elsewhere. In the upper left corner, you see a zoom in of an herbarium specimen collected April 28th, 1900, so 123 years ago, just outside of Pittsburgh. And when you look closely, you see the little yellow flowers, but no leaves. And this species flowers and then produces leaves, this shrub. But then 117 years later, as part of this kind of larger project, actually inspired by an exhibition going out back to sites where former botanists have collected and doing recollections and kind of doing these phenological comparisons. And here you see the flowers are actually long gone already, even though it's the same calendar date. And the leaves are fully flushed. And again, about the same population, same area, just outside of Pittsburgh. And this was featured in a museum exhibition here at the Carnegie Museum called We Are Nature, Living in the Anthropocene, where we had these kind of before and after comparisons of herbarium specimens. And this kind of launched a long-term project here at the museum by Bonnie, me, and others at the museum to go back to sites and do recollections, not only compare differences in phenology, but also differences in what makes up the flora. And the kind of the cool thing from this exhibition that I kind of took to heart was that these kind of before and after comparisons, though kind of one-off and oftentimes cherry-picked, were very kind of made climate change a little bit more real, locally real to museum visitors. And so that was kind of one additional inspiration to continue that. And then, of course, in addition to climate change, plant invasions or introduced plants also affect phenology. Here's a picture also from my backyard, or the woods behind my backyard, of Rosa multiflora rose invasion in the early spring. And as you see, forming very dense shrub layer, but the overstory canopy trees really aren't doing much yet. So you can see how the introduction of particular species with different phenologies might impact other native plants. So I broke the talk today up into kind of three categories here. First, talking about understory phenology and some of the basic work that we've been doing on forest wildflowers in the herbaceous layer, jumping into invasion phenology a little bit, and then ending with our kind of ongoing current work that's especially focused on climate change phenology. So starting with understory phenology. Really was kind of taken aback. My PhD work was really all on shrubs, in particular invasive shrubs. And I really didn't think about wildflowers very much at all. Didn't know much about them, to be honest. But kind of early in my first postdoc, I was kind of forced to start thinking about wildflowers. And now I'm all about wildflowers and kind of thinking about these spring wildflowers, or so-called spring wildflowers. How important is spring for spring wildflowers? And as a functional ecologist, I was kind of surprised that there wasn't really that many studies. We assume that spring is really important for these plants, but it wasn't really quantified or measured. And I got a question here. I've got the questions up asking, with flowers coming out early in the spice bush, how does that affect species? That spice bush is the critical host. And that's a great question. One I actually don't really have an answer for, but one that is definitely one that's huge for follow-up. I can say that there's a good bit of studies and an increasing number of studies on herbivory, actually using herbarium specimens showing the impacts of climate change causing earlier leaf out and being mismatched, either being mismatched or causing an increase in herbivory, and a lot of kind of crazy things that I haven't really studied, but I know other people have studied. And you can imagine that leafing out much earlier in some cases might actually help, depending on what the food source is. But in some cases it might help an herbivore, for instance, and there may be a better match, so to speak, or there could be a mismatch with the pollinators if the pollinators arrive when the flowers are long gone. So it really could go either way, and it's definitely species dependent. But yeah, so that's my answer of a non-answer. Getting the timing right is really important in spring. And so, of course, there's challenges of the risk of frost damage. Come out too early and you might get a late frost or just cold damage. And then, as the question just was asked, you might have a risk of ecological mismatch. In particular, we think about pollinators. Maybe if you flower at a time when the pollinators aren't similarly responding to climate, then you can have a mismatch. But there's a lot of perks too. So spring is a time of high soil nutrients and kind of a pulse of resources, not only nutrients, but also a high light availability. So kind of beating the trees and being able to get that high light in the understory before being shaded out. And then also just the earlier you come out, potentially the later you might remain and even have a longer growing window, which might also have advantages. And I like to just throw these pictures out, especially if you don't know the eastern deciduous forest or know some of these species, even like myself prior, thinking just about shrubs, because if you start your field work in April, you might miss some of a lot of these species that are kind of carpeting the floor, but then you come late spring, early summer, and there's little to no sign of the species. And so here's another charismatic Dutchman breeches, again, withered down to pretty much nothing in about a month's time. So there's a lot of different diversity of our forest wildflowers. So we have this kind of the charismatic spring ephemerals that come up really early and go dormant about the time that the trees leaf out. We have summer green species that leaf out a little bit later in spring and retain some green leaf tissue through the summer. And then we have evergreen species like hepatica shown here that may have leaves, oftentimes flowers in the spring, though not necessarily, and has leaves throughout the year. And so just showing this kind of more on a calendar of the different strategies, showing kind of the shaded area, kind of showing the pulses of activity among different functional groups of wildflowers. So evergreens have leaves all year round, but there may be particular pulses of activity at different times of the year, in particular spring. Whereas spring ephemerals are only around in that very short window and have a high level of activity in spring and then are gone above ground. And then summer green, which may have a little more low and slow strategy, but again, coming up late spring, early summer, and retaining leaves through. But the thing from all these different wildflower strategies, they're really defined by what's happening in the overstory. So here showing just kind of a little cartoon of overstory leaf index, or just the leaf out of the trees. Of course, in early spring, no leaves, and then super dark mid-summer, and then dropping again in the fall. And you can really just the inverse of that then is the understory light levels. And as you know, as you might know, it's super dark in the understory and in a lot of forests. And it's actually quite remarkable that these plants are living in such a dark environment. And in some cases, it's kind of the norm for trees to be blocking in an understory forest, in the understory to be blocking about 98% or more of the sunlight, which is pretty, so you can see that the early spring resource is pretty important for a lot of species. and in particular, summer greens, or in particular, the spring ephemerals, as you can see here. And so we might think of different species as either avoiding shade or tolerating shade, and different adaptations to live in this highly variable environment. And so spring ephemerals we might think of as, I like this term of phenological escape, kind of coming up really early and going away, and being able to take advantage of that highlight before being shaded out. And so again, this was some work that started in a postdoc with Susan Kalis, who was at the University of Tennessee, Knoxville, but doing work just outside of Pittsburgh, not too far from where I'm sitting right now, in fact, at Fox Chapel, at Trillium Trail in Fox Chapel. And this project was a really fun project with a team of students. And what we did is we looked at we kind of really closely studied 7 different common wildflower species with different phenological strategies, really basically asking this question of this seasonal kind of how these plants are taking in sunlight, photosynthesizing throughout the year. And so it really was a time to kind of get to know these plants and look at them individually. And here's just, we were able to look, all throughout the year. And we did some intense monitoring, measuring photosynthetic rates, in particular carbon gains, the amount of carbon dioxide taken in by the leaves, which was an intense effort. And some of these leaves are tiny. And in the end, it was really kind of a year-long campaign, a really intense kind of year-long campaign with many, many thousands of measurements to get what I'll show you here in a second. And a lot of this was made possible by a particular undergrad, Steve Cassidy, who I wanted to shout out, who is now a graduate student at University of Florida. And so we were able to measure, I'll just kind of throw some data at you, and then hopefully it'll make sense here in a bit, but just measuring a bunch of all these different species throughout the year, and looking at their responses to different light levels and to different carbon dioxide levels. And that's what I'm showing here. And this is just kind of like all of the data we've collected. Do some fancy modeling, use photosynthetic models throughout the year, and be able to incorporate here the light levels. So being able to understand how these different individuals are responding to different light levels at different points throughout the year, measuring light levels continuously in these plots, and then being able to model photosynthetic rate across the entire year. And again, light levels are super high in the spring, showing here in the understory. And then once the tree canopy closes, not surprisingly quite low, but maybe surprisingly is like super low, quite low. And then even after the release and the fall, a little bit of bump of increase in light, of course, with leaves that drop, but not nearly to the extent of spring. And so here's the species in particular we studied. And here showing in the background is these light levels that I just showed you. And then these horizontal colored lines are showing the leaf duration of each species. And then the vertical colored line is showing where, when across the season, the species on average flowers. And so you can see there's quite a diversity in both the duration and the timing of duration of leaves and the timing of flowering. But what was, I don't know, this maybe is nerdy, but hopefully some people find it interesting, because I really love this study and being able to look at actually model daily carbon gains, so the amount of photosynthetic activity that's happening every single day across the year, and being able to integrate that to understand how important are the different seasons to these different species. And you can see some variation. So most obviously spring beauty in the upper right in the orange does all of its carbon gain, all of its photosynthetic activity as a spring ephemeral before the trees wake up and leaf out, versus other species, maybe like false Solomon seal or Virginia waterleaf in the lower left, these summer greens that are doing a lot of photosynthesizing in the early spring, but also maintaining a relatively lower, but maintaining photosynthetic activity nonetheless through the year. And so the big take home from here, which is interesting and important in its own right, but I'll in the next part of the talk, you'll see why in particular quantifying this is important. But many species acquire more than half of their carbon, so do half of their photosynthetic activity before the tree canopy closure. So that's what I'm showing here circled. So here's kind of a cumulative plot of the percent of carbon gain each day, kind of adding up eventually until it reaches 100% of its carbon gain. And you can see kind of its annual or seasonal trajectory. And that dashed line is the point at which half of the photosynthetic activity for the year is reached. You can see for almost all, except for two, almost all of the species, that's reached before the tree canopy closes. And that's kind of surprising because it's even for those summer green species, some of those species that do retain leaves later. And so that's what I'm showing here. And then also it's also these species continue photosynthesizing also through the summer and acquire in some cases, relatively high proportions of their carbon gain. So in the case of the green jack in the pulpit, whereas relatively lower in some other species like Trillium, but still almost a quarter of its carbon gain happens in the summer for Trillium, which is perhaps not expected because we often think of it as a very spring species. And even more kind of intriguing is that fall does matter for some herbaceous species as well, in relatively high proportions. So we're talking like 20% of the carbon gain actually happens after leaf canopy autumn leaf fall. So just to summarize, spring is critical. About 39 to 100% of annual carbon gain in the species we studied happens in early spring before the trees. But summer and fall is also important depending on the species, of course. And another kind of interesting thing from this study is that these kind of discrete bins are kind of useful when we talk about different species, but each species really does kind of have its own strategy. And we think of these phenological strategies like spring ephemeral, summer green, really as being more of a continuous, should be really thought as more of a continuous strategy rather than one that's discrete that fits into these neat bins. I've got a question if there's any impact to soil moisture on these results. Probably, and that's something that's actually less studied for phenology in general, is the impact of precipitation at least in temperate deciduous forests. But undoubtedly, and so oftentimes we think just about spring temperature in a lot of these things, but we do think precipitation probably is important and is definitely important for like the duration of some of these phenological strategies. But it's largely kind of unstudied and kind of like the frontier now of phenological work. But in the case of these results, there really was no impact of soil moisture in that we actually did this study in what I call an in situ common garden, basically like individual plots where each plant roughly receives the same environmental condition. So yeah, so we were able to not worry so much about fine scale differences because they were kind of randomized in a plot setting. And then last, the understory phenology is actually all about the overstory. And I'll talk a little bit more about that in the climate change section. And then this section is real quick. I just kind of wanted to talk a little bit about invasion phenology, which is another large part of the work that we have done and are doing. And thinking about our introduced species, do they have different phenologies? And some past work that I've been involved with and some that I haven't been involved with has focused a lot on invasive shrubs in particular in Eastern North America, which makes sense because we have a lot of invasive shrubs in our native areas, in our natural areas. In some previous research, invasive shrubs tend to leaf out earlier in the spring and or drop their leaves later. So they have this kind of extended phenology. They tend to take high advantage of these higher light levels in spring. Paper by Jason Fridley over a decade ago now found that actually some species, a lot of invasive shrubs retain their leaves later into fall and can acquire a lot of their photosynthetic gains in late fall, which is surprising. Invasive shrubs have high resource use efficiencies, so they're adapted to disturbed forests for sure, but they also can make do quite well in relatively lower and more efficient use of resources. And yes, that's what I said there. So we know invasive shrubs are unique, but what about invasive wildflowers in particular in the understory? And so I'm guessing some of you thought I'd talk about this, and that is garlic mustard, widespread across eastern US, a lot of research on the species, allelopathic, so produces chemicals that affects below ground mycorrhizal networks in particular. Deer tend to not eat garlic mustard, and it has an interesting kind of biennial strategy, which I'll get to. And it has this extended phenology as kind of in its very nature of having this biennial strategy that in year one, it germinates in the spring or thereabouts, forms a rosette for a year, and then in its second and final year flowers and dies. And at least in my region, though introduced to North America much earlier, it really is a relatively later introduction. And so here's a cool specimen from our herbarium, one of the earliest for our region from 1956, actually collected at our study site, which is kind of cool. Specimen itself actually kind of looks like crap. So I like to think that it, I like to tell the story that this made up story, but maybe true, that the curator of botany at the time, Leroy Henry, was walking at Trillium Trail, saw this kind of unknown or unique species, grabbed it, put it in his pocket, then went back to the herbarium and noticed it and kind of keyed it out as garlic mustard and was like, ooh, that's interesting. And then plopped it on the sheet, and that's why it looks like crap. But I don't know if that's the case, but still we have very few specimens from there, and it certainly was a unique, very early collection. And so just putting garlic mustard on the same, excuse me, putting garlic mustard on the same kind of plot that I showed before with the native wildflowers, you can see that it's quite a different strategy with this biennial strategy. So the lower darker red garlic mustard year 2 dashed line, you can see it germinates in May, hangs out, and has a very long above ground, has above ground leaves, and then flowers the next year and retains photosynthetic activity through midsummer in its second year. And so putting it all together, you see that it has quite this kind of year 1 and year 2 biennial strategy is quite different from a lot of the native perennial strategies. And in particular, so again, showing this cumulative graph, in its first year, it kind of has a summer grain strategy, or similar to the summer grains. And in fact, it's about a third of its carbon, its 2-year carbon gain, about a third of its photosynthetic activity happens in the rosette stage, and the remaining 2/3 happens in its second year. And it very much acts as the spring ephemeral, is kind of like there with Claytonia in terms of rates of photosynthetic activity and timing. And so in many ways, it has a very kind of unique strategy. We do have biennials in our native flora, but relatively less common. And so in this way, garlic mustard achieves both this high photosynthetic rates throughout the year, and it kind of has this unique phenological niche. All right, the last part, which is, I think, the most exciting part, hopefully, you agree, is this kind of thing, kind of putting it together, invasions and climate change together, and thinking about kind of the future of our forests in this way. So we know that spring is a time of change, but spring as we know it is also changing. And so in the last section, kind of thinking about how we know different species have different phenological strategies. Do they also have different phenological responses to change? And so it's a big area of research, this idea of phenological mismatch. And this is defined as when species respond differently to climate change. So when interacting species respond differently to climate change, leading to an asynchrony. And oftentimes we think of this as, in the case of kind of as for plants, we think of plants and their pollinator. But there are many other different kinds of phenological mismatch, both known and unknown, that are possible or are happening already. And so this next part really inspired our current ongoing research, which I'll kind of overview. but this was a project that started a couple of years ago that led in some interesting directions that I really wasn't expecting. And now it's kind of a lot of what I study, which is exciting. That's thinking about mismatch between different forest layers. And I'll explain that in a minute here. And this was with some really fun people I was able to work with, including Susan Kalis, who I mentioned before, Richard Primick, Caitlin McDonough-McKenzie, and Jason Fridley. And so this really started in the last project when I was doing these kind of hardcore kind of basic measurements of forest wildflowers here in Pittsburgh. At the same time, and much earlier than me, Richard Primick was actually doing research collaboratively with Henry David Thoreau. And so he was going back to, so Richard Primick is at Boston University as a climate change ecologist. Who is doing a lot of really great things with phenology in particular, and was kind of among the first to kind of look at Henry David Thoreau kind of as a scientist and look at the data that he had collected over his really kind of almost 2 decades of observations of dozens of plant species and other organisms. And so he kind of was rewalking the footsteps of Thoreau, kind of seeing, to do these comparisons, use this historical Thoreau data set in a really interesting way. And so again, at the same time, I'm also doing this kind of very fine scale research, not in that lens, but looking at forest wildflowers. And so we kind of got connected in an interesting way here. And so we ended up looking at the Thoreau data set and combining it with my data set, which I'll show here. And so here I'm going to show I blocked it out so it's not too overwhelming to start. But on the y-axis, on the vertical axis, looking at leaf out date of different species. And on the x-axis, the horizontal, looking at the mean spring temperature. And on the top, on those box and whisker plots, those box plots, you can see the range of data, the range of spring temperatures across the different eras. So the era of Thoreau in the 1850s, another naturalist around Boston, Hosmer, and then also Richard and his lab. And so you can see in the Boston area, there's been about a 3-degree increase in spring temperature on average, 3 degrees Celsius. And after looking at this data set, it really became it was quite striking actually that trees and wildflowers were responding differently. So here in the very dark black is the trees. And in the dashed lighter gray are wildflowers. And the slope of that line is significantly different. And that's really quite important. So this is looking at about 15 species of wildflowers and about 14 species of trees. So on average, as the spring temperature gets warmer, trees are responding by leafing out earlier and at a much faster rate than that of the wildflowers. And so just to put some numbers on that, so trees since Thoreau's time are now leafing out about 2 weeks earlier, which is pretty wild on average. So not every single species, but on average. Whereas wildflowers are leafing out earlier, alarmingly earlier or interestingly earlier, but not nearly as much. So not even just shy of a week earlier. And so this is really significant. This jumped out at us as being not only is this phenological advance happening, which was already shown before and for many different species, but this difference between the overstory and the understory is significant because that critical window of spring, that high light in the spring that a lot of different species are adapted to take advantage of, is decreasing. And from our data set has decreased by about a week, which is pretty significant. But it doesn't matter. So this is some of our current work is kind of looking at, what are the implications of this decreasing spring window? And luckily, we did a bunch of nerdy photosynthetic work at Trillium Trail outside of Pittsburgh on a group of similar species. And so what we were able to do is actually model, do some simulations to model, well, what would happen if spring is getting shorter. And so that's exactly what we did here. I don't need to show you the math too much. So we were able to simulate and kind of model what spring energy budgets or photosynthetic budgets were 160 years ago, and then project to the future. So here, these different colored lines are showing the different species that we looked at. And on the top, you see the change in spring temperature. So we know that about 3 degrees, that the Boston area was about 3 degrees cooler 160 years ago. And then also the change in the spring length, defined as the days before canopy closure, so that the timing between the understory leafing out and the overstory leafing out. And so we're able to model the proportional change in spring carbon gain among the different species. And so from this, we can see that spring energy budgets were larger 160 years ago. So this change has already happened. And in Concord, that almost, depending on the species, ranges from 12% to over 25% larger 160 years ago than today, versus Pittsburgh, just as another reference, because that's where we did this other work, which had a smaller degree, had only about an increase in about 1 degree C over the last 160 years. So didn't have as large of a change as Concord, but still nonetheless, depending on the species, up to 10% different in the carbon budget. And then what's more interesting is kind of projecting to the future, given different IPCC climate change projections of what temperatures might be depending on various emission scenarios, that these spring energy budgets will keep shrinking. And so here are these B1, A2, A2, mean, max. These are just different scenarios, different modeled scenarios of change in spring temperature. And so it's showing the same thing, but forecasting to the future. And again, depending on the species, so here's bloodroot sanguinaria, could decrease by as little as 10% over the next, however long 2080 is, next upcoming decades. Whereas Jack in the Pulpit could decrease its spring budget by almost 50%, depending on the scenario. All right. I'm clicking. So just as a little summary of the kind of this past work is, perhaps not surprisingly, but interestingly, species responses differ. Trees are more responsive than wildflowers. And these mismatches have caused and will continue to cause declines. But in what way? Decline in the spring energy budget, how will that actually manifest on the landscape is less clear. And so that's what a lot of our grant and current ongoing work is looking at, kind of putting it together and also incorporating the impacts of invasions in addition to climate change. And so this is a collaboration with Richard and with Sarah. And we kind of have three big questions, and that is, what are the impacts on reproductive growth and survival? So we did some modeling, but we didn't really actually measure the impacts directly. What's the mechanism, which maybe is a question some of you have, like why are trees different than wildflowers? And then also in 3 there, biogeography, is this mismatch between the overstory and the understory, is that just kind of a quirky thing that happened where Thoreau was looking, or is that a larger scale thing across Eastern North America and in other temperate deciduous forests worldwide? So just to overview some of this ongoing work, what are the impacts of the early shading? So we have some ongoing experiments now looking at shading to mimic do artificial shading with shade cloth out in the woods to mimic early tree leaf out, what we expect the canopy to be the timing of the canopy leaf out in 2080, for instance. And then also simulating invasive shrub shading, which I'll mention here in a second. And so this work was largely launched forward by Jess Petit and Cheyenne Moore, who were at University of Pittsburgh at the time. Also looking at not only doing the artificial shading, but also measuring wildflowers under different shrub canopies that have different phenologies, so both invasive and native, and seeing what the impacts of these different timing of being shaded out and what the impacts are of that. Because like I mentioned before, a lot of non-native shrubs not only do they leaf out earlier, as shown here, but they also form much denser canopies. So being a trillium or another wildflower under one of these native versus non-native shrubs could have a huge difference in their function. Here's just some data from a couple of years ago from this project. And what you can see is on the top, this is the understory light level. You can see just comparing to a common native spicebush again, that a lot of some invasives really reduce the understory light level a lot much earlier in the season compared to spicebush. However, also shown here is giant knotweed, which is a whole nother area of our work. But knotweed, this species actually has a relatively later phenology. And so not all invasives necessarily have the same phenology. And then this also inspired other side projects looking at dozens of individually tagged plants. And this is a student-driven project by Abby Yancey, and just kind of throwing it out there. If anybody's going to the ESA conference in Portland next week, is it? I don't know. I'm actually not going. But Abby will be there, and she's giving a talk on some of her work on the side, which I think might be really interesting to the group, which was really driven by a natural history observation related to our project, but really independent. And that is noticing, here's just the exact same calendar date, March 29th, seeing two individuals just less than a meter apart, having a very different phenology, all because of the leaf litter depth being different. So she did a really cool leaf litter manipulation experiment. And so she'll report some of that at the ecology meeting, and hopefully we'll get that analyzed and written up too in the future. Yeah. And then I wanted to just address questions now too, since we're, so what's the impact of temperature increase in native plant meadows like it is to flowers in forests? Yeah, that's a really great question. We actually did some herbarium work. I actually don't have it in the talk, but we did some herbarium work in old fields in Pennsylvania, looking at the impacts of climate in old fields, in particular native and non-native plants. I actually didn't share that link with Maura, but I certainly can. But I would envision it would be quite different. And in particular, there aren't this kind of spring ephemeral strategy, at least to my knowledge, isn't really manifested in kind of prairies, open field settings. So I do think that there's some really interesting differences, and in particular, for some of the later blooming, how will they be affected by earlier season changes in climate, both precipitation and temperature? So there is research on that, some of which I've been involved, but I'm not. All I can say is that's a great question. In Minnesota, recent springs have been later. In 2018, a park where I volunteered had a foot of snow, April 15th. Park records indicate later appearance of ephemeral wildflowers. My impression, though, is that the blooming is also shortened, only a week for the rare dwarf trout lily. Is that a threat to their survival? So yeah, I guess my gut would be, yeah, that does seem like that would be a threat. That is really interesting with the differences in snow. And I think that the snow is definitely like the snowpack, which there's really not a consistent snowpack in where I am here in Pittsburgh, but there is in other places across the range of the same species. And a lot of Canadian research in particular, really, and alpine research and Rocky Mountains, for instance, really hits home on the importance of the snow melt date and how that's important. So it's really like kind of depends even within a species on the range what the driver is. So that's really, yeah. I'm curious about the fact of leaf litter buildup for spring ephemerals and how it might be different in the past under indigenous land management. That's really cool. I had actually never thought about that. It's a relatively newer project. Really haven't thought about that. We certainly are looking at like the different litter on different overstory species and how that impacts. Like some litter is more recalcitrant or more sturdy, slower to degrade versus more higher rich litter. But yeah, but with the burn histories and just forest composition generally changing as a result of land management, that's, I don't know. I'm not really answering questions other than, wow, that's really cool. In addition to invasive shrubs, have you been thinking about impacts of invasive trees? Yes, we have a little bit, and we looked at some invasive tree stuff, but not so much. And mostly because, dare I say to this group, there aren't a lot of dominant invasive tree species, at least in our area. There are in like urban areas and some urban parks and things, but not particularly in our study site. So that hasn't been a focus, but it certainly could and should be in a lot of different areas. Yeah, and did we look at the role of soil temperature plays versus solar exposure in the emergence? Yes, a little bit, and I have a little bit more on that here to come, I think, hopefully. And actually in this section here with the mechanism. So we've been doing a growth chamber experiment. So looking at what are the actual phenological cues. And for this work, we've been doing it at a site outside of Pittsburgh, owned by the Audubon Society of Western Pennsylvania, and been using actually some native plants that they have been propagating and have in their native plant center. And so for this, we actually did look at different temperatures in the canopy, in the tree canopy, shrub canopy, soil surface, and below the soil. And we also planted out a bunch of different plants in pots, or in the case of trees that are already there, and brought them back at different times throughout the year to be able to ask this question of what we think is that that the different functional groups, trees, shrubs, and understory wildflowers are actually responding to different cues. And so here I have highlighted, in the overstory, plants are responding to winter chilling, potentially day length, and especially air temperature. So temperatures in the air versus in the understory with their below ground buds, we suspect that they would be more responsive to changes in soil temperature in particular. And so we kind of are hypothesizing that soil temperature is buffered and quite different, less variable than air temperature. And some of our first data, show this. So shown the line in the brown line in the middle, you can see it's much less variable than at the other strata. And you'll see that in the winter, it's buffered, so relatively warmer than the air temperature. And then in the summer, it's cooler than the air temperature. So it has this kind of buffering effect and less variable. And so we brought these plants back in to the growth chamber at different times of the year, at different times through the winter, and put them in different environments. And really the take home here is we can look at, and I'll just hopefully this isn't too overwhelming, but this is looking at basically bringing in cut twigs and bringing in potted plants at two different times in the dormant season in January and February, shown here. And then just putting them in a warm chamber and a relatively cooler chamber, and then just seeing how long it takes for the plants to leaf out, or in the case of the wildflowers, emerge in their pots and leaf out. And so what you can see here is the slope of these lines and the differences between the different points are significant. And it's kind of a lot to grapple here, but what I just wanted to point to you here is non-natives are responding to spring warming, meaning that when you put them in a warmer chamber, they leaf out earlier, and they leaf out earlier generally compared to native plants. Whereas the native plants are very slow when you bring them in early in midwinter. They're very slow to emerge in January. But if you give them a little bit more time in the cold, then in February, they're much more responsive and come out. So they have a more conservative strategy. Yeah, and then someone asked about, does Pittsburgh area have non-native earthworms, reducing forest leaf litter in many forests in the Northeast? Your student's photo shows a healthy leaf litter layer. Yes, there are forests that have, that definitely are invaded by non-native earthworms. One that that's a great question. And that's like another one of these like other things. Like, so I haven't mentioned a lot of different changes. There's so many different changes happening in our forests. And that is one of them. And that's one of them, sadly, that we haven't been thinking about heavily. But like you mentioned, in our forests, I guess I don't know for sure, but it definitely has a very healthy, thick litter layer where we're studying. But that would actually be a really interesting, again, something I didn't thought of. That's great. Really interesting implication, perhaps unstudied, understudied implication of a different leaf layer depth, and that as a result of earthworms, and how that might affect some native wildflowers emergence times. So that's cool. I'm inspired. All right, and then the last part is herbarium specimens. Well, we've been looking across the world basically to see if this pattern of phenological mismatch has been held across, if it's just a goofy thing that happened in Thoreau's neck of the woods, or if it's in other woods as well. So this project was led by a graduate student at Boston University who now has a Ph.D. As part of their dissertation, Tara Miller. And this was recently published in Journal of Ecology. And it's kind of a lot here because it's both a comparison of this phenological mismatch between the overstory and understory, but also the differences between trees, shrubs, and wildflowers, and native trees, native shrubs, and native versus non-native trees, shrubs, and wildflowers. So it's kind of a lot. But perhaps the most top line interesting thing is that we did find that the trees are more responsive to climate across the range, although it differed at different points in the range. But overall, the pattern that we found in the Boston area was also found in herbarium specimens, that natives tend to advance out. Just thinking natives alone, they advance their leaf out times much faster in warmer areas, so particularly in the south versus the north. And non-native species are more responsive generally than native species, which also does match other work by others and the growth chamber study I just mentioned. And so we also took this, using this incredible, as more and more herbarium specimens are going online. Ben Lee, who's a postdoc here at the museum, in collaboration with other collaborators across the world, we're able to look at the herbarium record and see if this pattern is it unique to Eastern North America, or is it also shared in Europe and East Asian temperate forests. And so we looked at almost 7,500 specimens across 36 species across this range. What Ben was able to do, which is really cool, is using the herbarium, using this phenological data from the herbarium record, being able to estimate based on current climate what the timing on average of the overstory, the top three figures, and the understory, the bottom three figures for Eastern North America. And so they're kind of shaded depending on the day of year and the average leaf out dates. And so from that, able to look at, basically subtract the overstory from the understory from the overstory, to look at that window, that critical window of light before being for the wildflowers before being shaded out of the trees. So the difference between the tree overstory leaf out times and the understory leaf out times. And so as you can see, the different areas are different colors. So we see you might need to squint a little bit, but in Eastern North America in the far left, you can see that there is kind of a gradient. So always negative. So this is a future projection that this window will be decreasing. And this is especially true in the further north you go. But you see that the opposite pattern, see the pink, the opposite pattern. So the spring window light is actually the opposite direction, which is kind of curious and definitely requires follow-up. We don't have a great explanation other than speculation on different functional strategies and climate histories of the different regions, but it's really all just speculative and kind of like, oh, this is interesting. And then last, before I kind of finish, is I just wanted to give a pitch on herbaria generally, which I could totally give a talk, which I love giving a talk just on all of our herbarium work, which takes us from many different areas of research, both phenology and otherwise. But also just like basically that last half of the talk was really a result of looking at herbarium specimens differently, which many people are now, and thinking of herbarium specimens as phenological data, for instance, but they also provide many other really interesting and important insights, both for research and for applications. And here I'm just showing an increase in the number of studies that are using herbarium specimens. So yay. And some of this, and just really quickly, just mention, just kind of brag / mention some of the cool other things that we're doing unrelated to phenology with herbarium specimens here at the museum. And that is a couple of years ago, looking, and we have a current ongoing project led by postdoc Ben Lee, but looking at the that dirt and roots of on herbarium specimens are often not used, but they really have some really interesting information in there. And we were able to extract mycorrhizal DNA, fungal DNA from trillium roots dating back to the 1880s. So we're following up on that now too, to be able to look at kind of fungal plant associations, not only across their range, but across time. So there'll be more on that, hopefully. And then also looking at changes in plant invasion, so in their traits, in addition to their phenology, but also looking at changes in their traits. Here's Japanese knotweed in southwestern Pennsylvania, showing relatively, I mean, kind of striking evidence of a change in leaf size over the last 100 years in our region. So yeah, and so another study looking at trait changes, looking at stomata changes, and also looking at chemical changes in poison ivy, which was recently, will be out soon, which is a really interesting study and use of herbarium specimens. And then last, I just wanted to point out, for those of you that might be new to herbarium, which is that a lot of the work that we've done here was also made possible from the National Science Foundation grant a couple of years ago to digitize a lot of our specimens as part of the Mid-Atlantic Megalopolis, which is a group of more than 10 herbaria in our region in the Mid-Atlantic, digitizing and getting online. And kind of the biggest resource now is like, there's this huge resource for research and otherwise, and you can search our collection at midatlanticherbaria.org. And I think that link will be in the notes or sent out with the YouTube. And so there's just a lot that can be done. There's a lot that has been done with herbarium specimens, a lot that can be done now that they're increasingly being digitized. So please collect vouchers. That's my other little pitch, is not only are collecting vouchers important to document what is growing where in natural areas and otherwise, but also it enables future research in ways known and unknown, and is definitely worth the effort, both for the scientific method and for enabling future science. And if you're, I also want to give a pitch for some of the work we're doing at the museum, connecting iNaturalist with our specimens. So essentially using iNaturalist, this community science free platform, to be able to connect pictures we take in the field and the iNaturalist platform with our specimens. So we know, and you can read about that in this paper too, which we hope is a relatively kind of user-friendly, especially for maybe people that haven't collected voucher specimens in the past, or to make it more easy to be able to use iNaturalist to even make the labels for your specimens, in addition to having other information about the specimen, including pictures from the field and other plants and otherwise that are around the plant that was collected. And also I wanted to give a pitch for the National Phenology Network, and in particular an app, Nature's Notebook, or an online resource. And at your site, we've set up a site at our field station at the museum, Powder Mill Nature Reserve in the Laurel Highlands in southwestern Pennsylvania, that Abby Yancey, the student who I mentioned with the litter project, launched. And so far it's been really successful, and it's been a great resource, not only for enabling science, so having visitors being able to upload observations, phenological observations, but also a big, just a good point of connection with visitors and outreach, and having people being excited about phenology and seasonal change. So yeah, you can read more about that. I'm a big advocate of and a growing fan of National Phenology Network, and these what are called these local phenology programs that may be applicable in your case, just like it was for us. And then they make these really cool maps then, and you can, let's see if I can get that to go or not. But essentially tracking the progress of spring based on these kind of volunteer driven observations is really cool. So here showing the long-term difference in spring for this year, if it's an early spring or a later spring. And so there's been a lot of other research using these data in addition to these kind of interesting and important predictive maps. And then last, I just wanted to give a pitch that I'm excited that we'll be doing another webinar on January 16th with Natural Areas Association on a brand new project that we just got funded with generous support from the Richard King Mellon Foundation, where we will be doing many things related to invasive plants, but in particular, how we communicate about invasive plants. And we'll be making an exhibit and some educational outreach materials. And I was really excited about the NAA connection and this talk in particular as a time to kind of get people's feedback and have an interesting discussion on best practices on effectively communicating about invasive plants, both in a way that is interesting and interesting to a general audience, but also in ways, in particular, ways that are inclusive and not xenophobic or in ways that are kind of a turnoff, and to be able to communicate kind of the ecological impacts of invasive plants and inspire general awareness and action. So yeah, if you could attend that, that'd be awesome. I'm looking forward to that. So thanks. That's it. If you left already, sorry, I went a little long. Sure.
So earlier leaf out of tree species will reduce the window for early spring, late winter prescribed burns, which remove leaf litter and open forest canopies. Can you speak about the relationship of your work to forest reforestation activities, especially prescribed burns?
These questions are great. No, no, but I can try. Yeah, that's what I was excited about this group in general, thinking about, you know, I've done a lot of this work kind of more in a kind of basic science context and not so much applied or to like boots on the ground kind of activities. So I really haven't thought about it too much, to be honest, but I think it certainly would be relevant, I suppose, if the timing of these things, and then also in particular if timings of burns are to be stumbling over my words, in the context of both what's happening in the understory and overstory, that prediction of that could be really important on the timing. So yeah, I don't know. You gave a really long question. I don't know if I.
No, you did. That was the essential. That was the essential question.
Yeah, because I didn't give a very specific thing, but yeah, otherwise, like, yeah, that actually would be the timing of tree leaf out and tree canopy leaf fall is critical there. Yeah.
Okay, so I did get one question. Where can you sign up for the January 16 webinar? The registration has not been opened yet. We're working on that as soon as Mason and I connect and we get what we need to get that up. But yeah, watch every early in the beginning of the month, if you're on our distribution list, you do get an upcoming events. It will be in there as soon as it's available. So if it's not in July, it'll definitely be in August. So thank you so much. We appreciate everyone being here. We did run a little long, but basically the content was terrific. It really was fascinating. Thank you for all the work that you're doing, and to everyone who participated, you know, all that you will contribute to continuing these studies. So thank you very much, and we'll see you for our next NAA webinar or event. Thank you, everybody. Thank you, Mason, once again.
Welcome. Thanks.